I'm going to talk in this lecture about white blood cells, also known as leukocytes. First up, I want to start out by pointing out a few general characteristics that all the white blood cells share. And in part, we're going to kind of contrast those white blood cells with red blood cells also. So the first thing I want to talk about is the generic function or general function of all white blood cells, and that is that they in some way, shape, form, or fashion help participate in an immune response. So they are cells that help provide immunity or function in some way with the immune system. Obviously, that's in contrast to the red blood cells, whose main job was to transport oxygen. Now, also, if we look at all of the white blood cells, unlike the red blood cells, all the white blood cells do have a nucleus. So the fact that these white blood cells have a nucleus also stands out, or lets them stand apart from the red blood cells. We can also take a look at the average lifespan. Remember, the average lifespan of a red blood cell was somewhere in the neighborhood of 120 days. But when we look at these white blood cells, many of them are going to last for only a few hours to maybe a couple of days at most. And that's because these white blood cells, usually when they're produced, they function very quickly. Now, I said usually they last only a few hours to a couple of days. That's because there are some special white blood cells that can last years and decades even. But those are the outliers. On average, most white blood cells only last a few hours to a couple of days. So, you've got their function, obviously, which stands apart from that of red blood cells. You've also got the length of time that they are able to stay alive, and the fact that they, all the white blood cells, that is, have a nucleus. And those facts are going to make them a little bit different, obviously, than your red blood cells. Now, when we look at the white blood cells, they are broken up into two major groupings. Those two major groupings are called granulocytes and also agranulocytes. So you see they use the terminology granular leukocytes versus agranular leukocytes in this chart. But for short, we usually just say granulocytes, and also agranulocytes. Now, let me tell you how those two categories come about. The fact of the matter is, all white blood cells in some way have granules. These granules usually contain a variety of different chemicals that help the white blood cells do their job. The difference is whether or not you can see the granules underneath the light microscope. Granulocytes have granules that you can see. Agranulocytes have granules, but you cannot see them up underneath the microscope. So keep that in mind. All these white blood cells have granules. It's just that some you can see the granules with, and others you cannot. Now, to kind of, get into the actual discussion about the white blood cells here, one thing that I want you all to be able to do is definitely be able to recognize descriptions of those white blood cells. So, to start here, we're going to talk about it, or give at least some general descriptions of these white blood cells. And in part, it'll depend upon the granules. In part, it'll depend upon the shape of the nucleus. And in some cases, it will depend upon the size of the cell. So we look at the chart here, we have the granulocytes. They are neutrophils, eosinophils, and also basophils. And we're going to describe each one of them. You'll need to be able to recognize that for the purposes of your quiz/test. And I'm going to give you the descriptions as I usually characteristically give them to you on one of those two items. So, to start with the neutrophils, usually I go back and I mention the fact that they have pink cytoplasm, and usually the granules will be fairly pinkish to kind of light purplish lilac in color. So there's your description as far as the granules / cytoplasm of a neutrophil. Pink. That's going to be your buzzword. When it comes to the nucleus, it should have a nucleus that has anywhere between 2 to 5 lobes. This lobe number can change, obviously. The older a neutrophil is, the more lobes that it typically has. Now, because the neutrophil nucleus can change the number of lobes that it has, sometimes neutrophils are also referred to as polymorphonuclear leukocytes. Sometimes we shorthand polymorphonuclear leukocyte to just polys or PMNs, using the letters there. Next up, let's talk about the eosinophils very quickly. With the eosinophils, you're going to have red to orange granules, so there's your color. red to orange granules, and a bilobed nucleus. A bilobed nucleus just means that the nucleus has two lobes. So, in recognizing a description of eosinophil, you should pick up that it has red to orange granules with a bilobed nucleus. Basophils have purple granules, purple to blue granules. And since the nucleus of all of these white blood cells is purple and the granules of the basophils typically are purple, the way that we usually say this is that it has purple to blue granules that obscure, and obscure is O-B-S-C-U-R-E, obscure the nucleus. And that just means it covers it up and makes it very difficult to see. Now, if you could see it, as you can see here in the chart, that nucleus would have two lobes, but the reality is that nucleus is obscured. It's covered by these purple granules. And I'll show you a very quick image here of a histology picture. And if you look at it, you'll see that basically you can't distinguish the nucleus of that basophil. Now, you see the purple granules, the purple nucleus, it all mixes together. So it becomes impossible to differentiate the number of lobes, realistically speaking. So, once again, description of a basophil is going to be white blood cell with purple to blue granules that will obscure the nucleus. Now, if we move into the agranulocytes, we're going to have two cell types here. You have lymphocytes and also monocytes. Description of the lymphocytes goes something like this. It is the smallest white blood cell in size, meaning that if we compare just how big it is to the others, this will be the smallest. And it has a large single nucleus, and then it has a thin rim of cytoplasm around that nucleus. So, once again, it's a large single nucleus with a thin rim of cytoplasm. Very small cell. Monocytes, their description goes that they are the largest of all the white blood cells. So they're the biggest in size. And they have a horseshoe-shaped nucleus surrounded by blue cytoplasm. So, large cell, horseshoe-shaped nucleus surrounded by cytoplasm. That's going to be your monocyte. So please keep in mind those descriptions. Like I said, you're likely to hear me phrase some of that descriptive, one of those descriptions at least, for you on your test. Now, another item that you'll need to work at memorizing for each one of these various white blood cells would be when their numbers elevate. From a clinical perspective, a very common test that's performed is called a complete blood count, or a CBC. And when we get that CBC back, one of the values that we get back is a white blood cell count. And knowing the white blood cell count can be useful because if it's elevated, it usually indicates that there's some type of immune response occurring. Well, we not only like to know that the entire count is up, but we also like to know the levels of the various white blood cells. And the reason is because if one of those levels is higher than another, then it helps us figure out potentially what may be going on as far as the immune system is concerned. Let me see if I have it. So, if we take a look at table 19.2 here, there's a little bit of information that you need to pull out. You do need to know or understand when the level, or what is indicated, at least traditionally, by elevated level of certain white blood cells. So, for instance, neutrophils classically, if their levels are elevated, we associate that with a bacterial infection. Now, there are other associations, but I'm giving you the ones that I definitely want you to keep in mind. So, neutrophils, their numbers are elevated. It indicates typically bacterial infections. If we move to lymphocytes, well, with lymphocytes elevated, lymphocyte counts typically indicate viral infections. For the monocytes, elevations in their numbers usually viral or fungal infections. With the eosinophils, two processes potentially: parasitic infections or allergic reactions. And with basophils, we associate elevated numbers of those with allergic reactions also. Now you might wonder, why do we have two items associated with allergic reactions? Well, the reality is that basophils carry the chemicals necessary to cause an allergic reaction, whereas eosinophils carry the chemicals necessary to essentially stop it. So keep in mind those situations in which you would see elevated numbers of each one of those white blood cells. Now, one other item that I want you to keep in mind about these white blood cells is the number, or rather their order, from the most to the least numerous as they would occur in a healthy individual. if you look at this chart, it's organized in that fashion. The most numerous white blood cell will be a neutrophil. If we go to the chart we just had up a moment ago, you can see neutrophils account for 60 to 70% of all white blood cells. Now you don't have to know the number, but you should remember that it is the most numerous. Lymphocytes would be next on that list. If we look at the lymphocyte percentage, it accounts anywhere from 20 to 25% of all white blood cells. Monocytes would be third as far as the most numerous. And you can see 3 to 8% is their count. Eosinophils would be fourth on our list, and they account for anywhere from 2 to 4%. And that leaves basophils last. And basophils are going to account for usually less than 1% of all your white blood cells. So the proper order from the most to the least numerous would be neutrophils, lymphocytes, monocytes, eosinophils, and then basophils. There's a little mnemonic that we commonly use. It goes, never let monkeys eat bananas. Never let monkeys eat bananas gives you the first letter of each one of those white blood cells in the proper order from the most to the least numerous. Additionally, you need to know, when we look at some of these white blood cells, some of them are going to work via a process called phagocytosis. Remember, phagocytosis is basically termed out as being cell eating in A&P1 and in general biology a lot of times. So some of these cells literally phagocytize or engulf other materials, other cells when they're active. And I do want you to know what those are. Your three phagocytes are going to be neutrophils. As you can see over here, they work via phagocytosis. Monocytes, they also work via phagocytosis, as it says here. And then we also include eosinophils, which have the ability to phagocytize antigen-antibody complexes, as it says here. Now, obviously, I don't expect you to know what an antigen-antibody complex is. We haven't talked about it yet. But because of their ability to do that, they can be phagocytes. Now, eosinophils aren't always phagocytes because, as I told you, they do release chemicals that help decrease allergic reactions. And you can see that this is potentially one of the things that they do. And when they do that, they usually don't phagocytize anything. They're just releasing chemicals. So their functioning as phagocytes is really kind of dependent upon what they're being used to do. Now, if we go back and consider the fact that blood is found within blood vessels, at some point it will become necessary for white blood cells to leave your blood and then exit into your tissues where there may be an active infection occurring. This process is called immigration with an E. Immigration with an E is the process by which white blood cells and blood leave your bloodstream, exit your blood vessels, and then will enter your tissues. Now, typically what happens to start this whole process is something called chemotaxis, C-H-E-M-O-T-A-X-I-S. Chemotaxis is a form of chemical attraction. Infected tissues, damaged cells begin to release chemical messengers called cytokines. And these cytokines will begin to attract the white blood cells to the infected area. So instead of floating through your blood normally and just kind of casually rolling or moving along, those white blood cells, in response to the cytokines, begin to make their way toward the edge of the blood vessel, a process rather known as margination. Once these white blood cells have marginated to the lining of the blood vessel, they begin to adhere and stick to it, and then they roll along until they find a little opening in between the cells lining the blood vessel, and then they will squeeze or immigrate out of blood and into the infected tissue where they can have their effect. So that's the process of immigration. You need to understand what immigration is, and like I said, it simply is the process that white blood cells go through in order to exit or leave the blood. And you should also understand what chemotaxis is. That's the chemical attraction that's going to cause those white blood cells to ultimately immigrate out of blood. Now, very quickly here, I'll also give you two terms that I want you to be familiar with. Those two terms are a leukocytosis and also a leukopenia. Leukocytosis and leukopenia are abnormalities in the white blood cell count. A leukocytosis refers to an elevated white blood cell count. It's not necessarily a bad thing, because a leukocytosis, if you're ill or sick, should be occurring. it means that your immune system is fighting, is trying to get a hold of whatever's causing you to be ill at that time. Versus a leukopenia, which is a low white blood cell count. A leukopenia typically is never good. A leukopenia usually means that you're losing the fight if you're sick, or it could indicate some other process like a leukemia could be occurring. So leukocytosis, elevated white blood cell count. Leukopenia is a low white blood cell count. And having discussed that, that'll pretty much wrap up our discussion here for the white blood cells.